Every cell in our bodies has an instruction manual called DNA, or deoxyribonucleic acid. It is what controls the cell but what also makes us different. The shape, which looks like a twisted ladder, is called a double helix because it is two strands that are linked and twisted together. When it is time for DNA to replicate, or make a copy of itself, there are special helpers in every step called enzymes that speed up the process since our bodies need to grow and heal daily, which takes billions to trillions of cells to do. First, the DNA is unwound and separated like a zipper. The original strands have their opposite pairs made at the same time as the unzipping until the DNA has been copied. The two new sets of DNA coil up immediately and are ready for when the cell divides. DNA is the material we pass on through generations, but what also controls the cells in our body. That means that DNA replication is essential to our survival. It all starts in the nucleus of the cell, where DNA is stored and kept safe. There, at special starting points called replication origins, an enzyme called helicase will start unwinding the double helix structure and cutting the strand of DNA to create a replication fork, so that DNA polymerase, another enzyme, can build the new half of each strand. The way DNA replicates is called semi-conservatively, which means that half the original strand is in the new strands. The old strand, otherwise known as the template strand, is used by DNA polymerase to build the new complementary strand. New nucleotides, which consist of a phosphate group, deoxyribosugar, and a nitrogenous base, either adenosine, thymine, guanine, or cytosine, are strung together oppositely according to the template strand, since DNA has an anti-parallel sequence, which means the strands run in opposite directions from each other. So if A was on one strand, T would be on the other strand, and vice versa for C and G. Once the two new strands are checked over by DNA polymerase to make sure that there are no mistakes, they coil back up and reside in the nucleus until cell division. DNA, the hereditary material in cells, undergoes a sequence of processes in order to replicate. Eukaryotic DNA replication starts in the nucleus of the cell and occurs during interphase so that there is an identical set of DNA for when the cell divides. DNA replicates semi-conservatively, which means one half of the original strand is used to build the other half, resulting in two new strands each containing half the original. The process starts from the replication origin, a special starting point that can have several locations along DNA, and a replication fork is created from helicase as it unwinds and separates the double helix structure. Special proteins called single-stranded binding proteins, or SSBs, block the exposed nitrogenous bases so that they don't rebond. DNA polymerase begins to synthesize the complementary strands from the template strands in a process called elongation. But first, RNA primers, which are 10 to 60 base pairs, are annealed or heat-sealed onto the strand, and act as a guide for DNA polymerase to start synthesis, since it's specially made to only synthesize a certain direction and needs an indicator on where to start. This direction is 5' to 3', which refers to the carbon backbone of the deoxyribose sugar in the nucleotides, where counting from the oxygen, the 5th carbon runs down to the 3rd carbon, and vice versa on the other strand. The carbons run 3rd to 5th. Adding on to that note, the nitrogenous bases of each strand are oppositely paired with their complementary base, adenosine with thymine and cytosine with guanine. The nitrogenous bases cannot be paired in any other sequence because of the special structures and bonding. Pyrimidines, which are T and C, only have one carbon ring and must bond to a purine, A or G, which have double rings. A and T form 2 hydrogen bonds, while C and G form 3, which is why the complementary bases cannot be changed. Overall, this sequence in DNA is called anti-parallel. The leading strand is the strand that is built in the same direction as DNA polymerase works, while the lagging strand is the strand that is built in small fragments called Okazaki fragments because it runs 3' to 5', opposite of DNA polymerase. These fragments are glued together by ligase, which creates a phosphodiester bond to link the nucleotides. When two replication forks are in near proximity, a replication bubble can occur, but this doesn't affect the replication process. Finally, DNA polymerase acts as a proofreader and makes sure the newly formed strands are correctly sequenced. If not, they cut out any problematic nucleotides and replace them with the correct ones. The two new strands coil up as soon as they are done to conserve space and are now ready for when the cell divides.